We will continue our discussion about the effect of VAM on the pancreatic cancer cells. In the previous video, we have discussed the VAM effect on the proliferations of this PDAC, that is, how these pancreatic cancer cells behave when the VCAM is upregulated or downregulated. In this video, we will discuss the effect on the migrations and invasions. In order to check this effect in vitro, the researchers did a wound healing assay where they have find out that when they knock down VCAM-1 both in the PANC1 cells and the KPN2 cells, they see the decrease in the migrations or invasions, decrease in the invasion of the pancreatic cancer cells that can be seen here. That is at 0 hour, the difference is almost same in both the PANC1 cells and the KPN2 cells when they use the control and the silencing VCAM-1 RNA. However, after 24 hours, what they have seen is that migration, this invasions in the PANC1 cells is in the control case is higher compared to the silencing VCAM-1 RNA, and same things they have observed for the KPN2 cells. And these things are been quantified for the multiple runs on the right hand side, where we can see that the decrease in the migration rate is significant both in the PANC1 cell and the KPN2 cells compared to the control case, indicating that VCAM helps in the migrations of the pancreatic cancer cells. And when they did the reverse, that is, they upregulate the VCAM in the PANC1 cells and also in the KPN2 cells using pcDNA 3.1 dash VCAM-1 plasmid, what they have found out is that there is increase in the migrations after 24 hours, both for the case of PANC1 and also for the case of KPN2. And these things are quantified on the right hand side, where we can see that for the multiple runs, the difference, there is an increase in the migration rate, and that is statistically significant for both the PANC1 cells and also for the KPN2 cells, indicating that within the VCAM is directly proportional to the migration and invasion of PDAC. VCAM is high, migration rate is higher, and VCAM is low, migration rate is lower. Next, they checked using the transfell assay, where the objective is to find out the invasion properties of the PANC1 and KPN2 cells. And when they use this silencing VCAM-1 RNA, what they have seen is that there is a decrease in the invasions of the cells. There is decrease in the invasions and also in the migrations of the cells for the PANC1 cell line and also for the KPN2 cell lines. And this quantifications of these results are on the right hand side, where we can see that in the case of invasions, the invasions of the PANC1 cell and KPN2 cells where this VCAM-1 is been silenced is lower, statistically significant, and is on the lower side. And same things they have observed for the migrations assay also. And when they upregulate or overexpress this VCAM-1, they find the reverse result, that is, there is increase in the migrations and invasions in the PANC1 cells and also in the KPN2 cells. And their quantifications are on the right hand side for the multiple runs, where we can see that invasions for the PANC1 and KPN2 cells is higher and is statistically significant compared to the control case. And same things they observed for the migrations. And there are some results they have shown in the supplemental figures 4, where they have provided the results of the experiment that shows that this VCAM also helps in recruitment of the macrophages that in turn helps in the leukocyte recruitment, and that results in the more increased lifetime of the pancreatic cancer cells. If you want to understand those, if you want to see those results and understand those experiments, you can go through the reference, where in that experiment they use Th1 cells of the monocytes and check that whether it will lead to the leukocyte recruitment at the site of at the site of this tumor microenvironment or near the pancreatic cancer cells. And I hope you understood this, how this VCAM modulates migrations and invasions using these main figures. If you want to understand these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.